Hi Nat5s, so the last section in this key area we're looking at the limiting factors in photosynthesis, so basically what decides how fast it can go. So just to remind you what a limiting factor is, because we've mentioned it in respiration. Okay, anything that decides how fast the reaction can actually go, that's a limiting factor. In an equation, you look at everything that's going in and anything that will affect how fast the reaction can run into the products, which generally is talking about enzymes. Okay, so photosynthesis has three. I've got, I think, enough space for me to be able to write this back in. So here's our photosynthesis equation, just to remind you. Okay, so we've got carbon dioxide and water, and we've got light, that's E with an L down the bottom if we're not clear on that one. Over the top we had chlorophyll, but we also know that there are enzymes involved in here. Okay, and glucose and oxygen is what we get out. So that's our summary equation. So basically our three should not surprise you. Okay, carbon dioxide concentration, okay, light intensity, not surprising, and temperature, because here we have proteins. Chlorophyll is not an enzyme, but it is a protein. Okay, so that has the same curve that you see for a normal enzyme curve for temperature. Now the reason that water is not in here is basically because you either have enough and you're okay, or the plant will die. So it's not going to limit the rate, it's just going to be you've got enough or you don't. Okay, so our three, this is the ones you need to know. So we're going to run through each one. Let's start with carbon dioxide concentration. Right, so it's kind of obvious. If you increase carbon dioxide, then you would increase photosynthesis, because the more carbon dioxide you've got, the more carbon you can fix in the carbon fixation stage. But it'll keep doing that until something else is the problem. So this is what the curve looks like, and this is what you need to know is the curve. So as you're going up, so as this carbon dioxide concentration is increasing, so you start at 0, and let's change the colour of that pen. Okay, you start at 0, you get 0 photosynthesis. Okay, you go up to here, you'll notice the rate of photosynthesis has gone up. I go up to here, it's gone up, and it keeps on doing this. As I go up, it goes up. As it goes up, everything's doing really well until you get to here. Now I've gone up, and I'm here. I go up again, and I'm still in the same place. I go up again, so I'm bumping up the carbon dioxide concentration. You're like, I should be increasing, but something else is the problem here. Something else is causing an issue with the rate. So you need to be able to recognize that. So it's quite simple. If you're looking at this one or the next one light, as you're going up the way, so as you're going up on the curve, okay, then whatever you're doing, as it's doing this, you look for the thing on the bottom, on the X. It will be the problem. But as soon as it starts going flat, something else. Okay? So your light intensity does exactly the same pattern. It's really quite straightforward. It's going to do, as you increase, it increases until something else becomes a problem. So the graph looks exactly the same, and it's exactly the same deal. As you're going up here, it's what's down here. But as soon as you start going flat here, it's something else. You don't know what it is, you just know it's one of the other factors that we know are potentially a problem. So remember, there are only 3 factors. So it's either your carbon dioxide concentration, or it's not light because we said it's gone flat, or it's temperature. So you just need to think, okay, which of these 2? If you're asked what could be the problem, it doesn't matter, either of these 2. Temperature doesn't look the same graph, but it's the same one that you've already seen, so hopefully it's not going to be too stressful. So it's going to do the same thing that you did for normally for enzymes. It's going to increase until we get to optimum, and then enzymes denature, so activity drops off. We have an asymmetric curve. Okay, so down here we've got our as you increase temperature, you increase rate thing, and that's just mainly because things are moving a bit faster, and so they collide more often, and then we finally reach our everything's perfect, wonderful, optimum temperature. Now remember, that temperature for us is like 37, but for plants that might be, 5-10 degrees depending on where they live. And then we've got this problem that's happened here. We have knackered our enzymes, we've denatured them, and please, please, please be careful. Do not say the word kill or die. Okay, so we've got our denatured enzymes. Okay, nothing else in terms of a term. And these enzymes have lost their shape. And if they've lost their shape, they lose their function. Okay, now this links all the way back to your enzyme stuff in Unit 1. Okay, so you can talk about active sites and things like that. The exactly same thing is happening with chlorophyll. If it loses its shape, it can't absorb the light that it should be absorbing. Okay. So the most complicated thing you could be asked to do is to analyse a limiting factors graph. But what's more difficult is they could give you several factors to get a complex graph. The simple ones would be if they just had one thing, like graphs I've just shown you in the last wee bits. Okay. But this is also possible. So you've got rate of photosynthesis. Let's take light intensity as being the one on the bottom, and you've got a curve. And you're like, okay, I know what that means. What you know is going up here. Let me get a wee highlighter one on this one. So up here, I know it's going up. So light intensity is the issue. And then I've got it going flat here. So I know something else is the issue. So if you were asked this, what's going on here, you could give points on the line and everything. It should be fine. But what if they gave you more information and they said, actually, this is a light intensity, but this whole graph has been drawn at 0.1% carbon dioxide and 15 degrees C. Okay, and then they add in a second line and they tell you that one is at 0.1% carbon dioxide and 25 degrees C. So you have to look at this a little bit more carefully, and you're still okay with saying, well, up here, that's light intensity. You know, that's the graph is going up. The problem is the thing on the X. Okay, so that's okay. But when you get to these ones, you can use the fact that you've got two lines to tell you something else. So here, the problem must be temperature. Because if you look at the one that's above it, okay, and this is the tricky bit, here you've gone from 0.1% to 0.1% carbon dioxide. So, but it's jumped up. So carbon dioxide can't have been the issue because you haven't changed the carbon dioxide. You have, however, changed the temperature, and that's jumped up from here to here. Okay, so it's gone up on this line. So 10 degrees temperature shifted the line up. So if they ask you what was limiting from here to here, you're like, well, that was temperature. Here, though, you have a problem. If they ask you what is limiting on this section, all the way along here, so I'm changing my colours and it's not working properly. There we go. Okay, so if we have this one here, you're like, well, I don't know. It could be temperature again. Like if I had another line that someone had drawn in for me, and the line was, say, running up here. Let's take it up here and ended up here. Oh, that's on the purple. That's not helpful. Right, let's take it up here. Okay, and I said that was 0.1% CO2 and 30 degrees. Then it definitely, yep, temperature is the issue. But if I had a line that went and basically it followed the same path as the second one, and I'd said 30 degrees, you're like, well, it's not temperature then, it must have been the carbon dioxide. So you would need more information. So just have a look at these ones that come up in the past papers, make sure you're happy with it. But the most important thing is the idea that if it's going up, it's the thing on the X, and if it's going flat, it's something else. And you only have three things that potentially could be there. And that's it.